Thank you very much for the introduction. Best regards from Jörg Line, who could not be here. And I also thank the organizers for giving me the opportunity to present this great hall. I would slightly change the topic. So it's advanced fluorescence microscopies, but I'm presenting metal-induced energy transfer, which is a recent technique we developed in our laboratories, which has exciting results we want to share with you. So it's known that, we already had the introduction that we have a resolution limit problem for microscopes. So if you have this abyss resolution limit, where even if you increase the numerical aperture or the collection angle of your objective, you cannot go beyond a certain number, which is lambda divided by 2 times your NA. In invisible light, it is around 200 to 300 nanometers, and it is not enough to resolve your biological moieties. So several methods have been developed, as was introduced in the previous talk. Single molecule localization-based techniques here. You take a frame of single molecules in each frame, and you localize the molecules based on the number of photons you collect. You have an accuracy. And if you sum them, you don't see anything. But whereas if you localize them from each frame to frame, you will resolve the distances and the position of these molecules. With the recent development of buffers and stable molecules, stable difluorescent molecules, you could go down to around 5 to 10 nanometers in the lateral direction, and you can resolve beautiful structures of biological complexes. For example, there's a nuclear pore complex, which we'll have a next talk. It represents, the proteins are labeled here with fluorescent molecules, and if you resolve them with the storm microscope, you can see the 8-fold symmetry of the arrangement of proteins in the outer ring. But this is not the end of the story here, because the world we live in is not a two-dimensional world. What we see here is basically a top projection of the complex, and it is known that there are 2 identical rings on top of each other, and you cannot. What you see is only one of the rings, or the projection of this complex. And in order to resolve the 2 rings, or separately, if you see the 2 rings, you need an axial resolution. A word about axial resolution. For conventional microscopes, it is much worse than your lateral resolution, which is in the order of half a micron to a micron. It is also true for a STED microscope, which does not improve the resolution in the axial direction. The palm and the storm methods, as shown in the previous slide, when you combine them together with astigmatic imaging, biplane imaging, or helical wavefront shaping, you can resolve or you can improve the resolution up to 50 nanometers. And there are also some intensity-based methods such as supercritical angle fluorescence imaging or total internal reflection fluorescence microscope methods, which can resolve up to 10 nanometers in the ideal conditions. But these suffer from intensity fluctuations or intensity variations in time, and you need to calibrate. For example, for this case, for every refractive index change in your medium, you will have some errors. The only method which could resolve down to 5 nanometers in 3 dimensions is the interferometric palm. So it has an isotropic resolution in 3 dimensions. However, it is a far interferometric, which involves the alignment of 2 high numerical aperture objectives on top of each other and 3 CCD cameras, which makes it technically a bit challenging to realize, and also restricts conditions on the biological samples that you want to measure, which is not so easy to apply this technique. Therefore, we wanted to develop a technique to improve the axial resolution of a microscope, and that's where the metal-induced energy transfer comes from. So before going into the details of the method, I would just go into a bit of fundamentals. So fluorescence is a 2-state process of a molecule. So you have a ground state and excited state. You excite using an excited laser, and the molecule it stays in the excited state for a couple of time before it comes down, either radiatively giving out a photon, which is fluorescence photon, or non-radiatively due to the collisions or heat transfer into the surrounding. So the average time the molecule spends in the excited state is given by the sum of these 2 rates. That's the average lifetime, is the property of the molecule. The way you measure this property is by using a pulse laser. So you pass a train of pulses of lasers, and you measure the photons and the time between the detected photons and the previous laser pulse. In this way, you probe the arrival time to the photons and you histogram them, which is shown here. And this histogram tells you the average time the molecule spends in the excited state. If you fit with an exponential decay, for example, you get the average lifetime of the molecule. Apart from this, another fundamental important quality of the single molecule, of a fluorescent molecule, is that it behaves like an electric dipole, which oscillates with a terahertz frequency, and it emits electromagnetic radiations perpendicular to its oscillation direction, as shown here. So if you look at the energy distribution in space, in a homogeneous space around this oscillation dipole, it follows the sine square theta law, where theta is the angle of the observation point and the axis of the dipole's oscillation. So it looks as a toroid in shape. So now this is the situation when you have a molecule in free space or homogeneous solution. Let us look at what happens when you put it on top of a glass surface or interface. So here I show two orientations, horizontal and vertical dipole, so as the arrow marks the dipole orientation. So before here, you have the refractive index below and above as same, and I'm cutting the toroidal section for just visualization purposes. Now if I change the refractive index below, you will see that the angular distribution of the energy of the radiation changes dramatically. So I'll pause it here. So this situation shows where you have air on top and glass below. As you can see, the shape changes. This is because of the tunneling of the non-propagating field from the dipole, which now propagates in the denser medium, and you observe such a angular distribution. So you use the dipole emits more radiation into the glass medium, and that's the reason why we use inverted microscopes now with high numerical apertures to get more fluorescence from the single molecules. As you change the distance, or as you bring the fluorophore close to the surface, close to the interface, you increase its radiative power. So you increase the radiation rate and you decrease the lifetime, which is shown by these curves. So this is the case when you have an air and glass interface. So here you see a change of 30% to 30% lifetime, depending upon the orientation of a dipole. The question is, can we do better? So let's take the condition, the situation where you have a thin gold layer on top of the glass surface. So the new thing here is that you have surface plasmons on the metal surface, as was introduced in the previous talk. So the dye molecules, they lose the energy to the surface plasmons, or the surface plasmons take the energy from the dye molecule and quench the fluorescence. So they indeed change the radiative rate again, and the lifetime curves now follow this trend. So if the fluorophore is directly on the glass, on the metal surface, complete quenching takes place, or complete energy transfer takes place, and you see no fluorescence at all. And with the distance, now you see a nice variation of the lifetime, which is an indication of the radiative coupling of the dipole to the metal surface. So this is the relation, this monotonous relation you can use to get the position of the molecule from the surface, as you can see. So this is similar to something which is known, the Förster resonance energy transfer, FRET, which has been used instead of FRET, where it is limited to 10 nanometers. Here you have up to 120 nanometers where you can observe this monotonous trend. This is exactly what we will use in the next couple of slides to resolve, to get the distance of the fluorophore from the metal surface. So we take a standard confocal microscope. So the confocal part is here, the pinhole, which projects the light out of the focal plane, and you have a cell, a living cell, which has a labeled membrane on top of this on the objective, and you can scan your beam to excite the different parts of the cell. The fluorescence is collected by a photodetector, which can time the fluorescence photons, and you can get the average lifetime information as a function of position. So the top figure shows the intensity image, and the bottom figure shows the fluorescence lifetime image of the labeled cell membrane. So now, using the curves in the previous slide, we can convert this this lifetime into height, which is shown in this figure here. So this is the height of the cell's membrane, the bottom membrane on top of the substrate. As you can see, the scale here is in nanometers. So now we can do this in time. And what we measured the adhesion of the early stages of spreading of a cell on the substrate. So we identified 3 stages. The first stage, where you have initial bonding of the addition molecules to the extracellular matrix. And so in the second stage, the cell membrane flattens out. And after that, it forms a lamellopodia, which spreads on the surface and increasing its surface area and decreasing its distance from the surface. The average distance of the cell from the surface is also a characteristic property. As you can see, so I take here 3 cell lines. The first cell line is an epithelial cell. The next 2 cell lines are carcinoma cell, cancer cells. And the average height for these 2 cell lines is higher than normal epithelial cells. And also, one important feature of the epithelial cells is that they have a wavy structure because of focal adhesion points, whereas the cancerous cells, they are comparatively flatter. Another application of this is we applied to the study of the epithelial to mesenchymal transition. So this kind of transition where the epithelial cells, they separate out So here you see they're ordered, they separate out and spread on the surface. They show a difference in their actin fibers or the stress fibers in the network inside them. And they express low amount of connecting proteins, E-cadherin, which is responsible for connections between the cells. This is an important process which takes place during wound healing or embryogenesis, for example. So we did the time-lapse MIET imaging on this process again. So we target the cells with the help of a transformation growth factor. And the cells, they change their average distance from the surface. And during these times, you measure the transition state 1, where they have a lot of dynamics going on between the substrate and the cell membrane. And later in the transition state 2, they form strong stress fibers, as was shown in the previous image, and they spread again. And in the last stage, they settle down. They come down to the untreated cell level, so they come down to the surface and do not spread anymore. So they're not motile anymore. So it was known before. They had theoretical, models of this, but it's the first time we are seeing such a proof, such an experimental evidence. It is nice to see that. So during the first stages, you have a lot of activity in the cell membrane. Here are the four time lapses which show the average height of the cell increases, and apart from that, you see a lot of structures which are dynamic and change a lot during the time. So I'm plotting here the line, the height profile from a line from these 4 images or 5 images, and what we see is the appearance of focal adhesion spots and the disappearance of the focal adhesion spots in some places. So during the first stage, a lot of focal adhesion spots are disappearing, and in the last stage, they are appearing again, so they come down to the surface. So far, I labeled only the cell membrane. We can actually directly monitor the cell's cytoskeleton by labeling it with the help of fluorophores. So here, for example, on the top row, I show actin of the cells. So these are mesenchymal cells which spread a lot on the surface. So here you see, again, the stress fibers. And at the bottom, you see a vinculin protein, which is found at the bottom of the focal adhesions. And as you can see from this image, the color scale represents the height. As you can see from this image, roughly, the vinculin stays at the bottom, whereas the actin changes the height along the fibers, which is what I show here. So if I plot the lines of three stress fibers along the length, and also the actin, so the vinculin and the actin protein labeled, and as you can see, the height is for the actin is higher and it increases. So you can also measure the height of your actin fiber that is forming this focal adhesion here. And after this is the data for 12 hours, and this for the 24 hours after the cell has been deposited on the surface. And you can also see, for some cases, you see that the distance between the actin and the vinculin also narrows down, whereas the vinculin stays at its position of 10 nanometers. By the way, this is a dual color experiment, so you label the actin with a different color and the vinculin with different colors. So that means that you can simultaneously probe different proteins and the distance from the different proteins from the surface simultaneously. So far, I have not talked about the errors of the estimation of the height, which is what I focus on this slide. So here what we are doing is we have, again, fluorophores on top of a silica spacer, which is transparent, and on top you have optical glue so that the refractive index is identical here. And we take lines of 100 pixels with different fluorescence excitation laser. So we have different counts, 1,000 counts per pixel to 5,000 counts per pixel for all the 100 pixels. And as you can see, the fluorescence lifetime shows a large fluctuation here if you collect only 1,000 photons, which improves down if you increase the number of photons that you collect. And this, again, translates directly to the axial heights that you calibrate from the calibration curves. And you can get a theoretical curve which shows that you improve or you go down 5 nanometers or 4 nanometers in accuracy in the Z, in the axial direction, if you have more than 5,000 counts per pixel, which is the advantage of the power of this method. Meanwhile, while doing all the cell measurements, we wondered if we could do also with single molecules. And that's where we designed some simple proof of principle experiments. This is again a confocal microscope. Here we have a pinhole. And what we are doing now is we are spin coating single molecules, individual fluorophores on top of substrate, on top of silica spacer with a predefined thickness. and we are measuring lifetime images. So as you can see from the color code, so the color code gives the lifetime, and you can see for different height thicknesses of 20, 30, 40, and 50 nanometers, you already see, if you can see, I mean, there's a difference of the light, color of your single molecules of the lifetime. If you histogram the lifetimes from each individual measurement, you see that they are widely separated from each other. So the lifetimes are separated for different heights, which means that you can already get the height information from each single molecule, each single fluorophore in your experiment. And if you take the maximum or the error bars here, the error bars correspond to 2.5 nanometers accuracy in the z direction. And the points that you collect nicely fit the theoretical calculations or the theoretical model, and the fit shows that you have 99% horizontal molecules on the surface, which is true for the case if you have smooth surfaces and the light molecules are lying flat on the surface. If you ask, this resolution is enough for resolving protein complexes, because proteins are usually in the order of 5 to 10 nanometers. So we tried measuring the lipid bilayers. So it's basically a cell membrane which you have two layers, the top and the bottom layer, which are labeled. And the height of this layer, of this bilayer, is 5 nanometers. If you use gold as a substrate, the lifetime difference between these two layers is less than 0.2 nanoseconds at the best, which you will not be able to resolve simultaneously if you record a point measurement. But we use something called indium tin oxide, which is a transparent conductive material, which has a very low absorption coefficient, because of which the lifetime curve, the calibration curve, is much steeper, and most of the lifetime modulation takes place in a couple of nanometers from the surface. So with that substrate, you can now achieve a lifetime difference between the bottom and the top layer of 1 nanosecond, which is resolvable. So we take a point measurement, and indeed the data that we take cannot be explained by a mono-exponential fit, but you need bi-exponentials, or two exponent components of 2 lifetimes. The 2 lifetimes already tell you the height of the bottom and the top leaflet. Therefore, we can achieve the thickness and the distance between the bottom leaflet and the substrate from such a measurement. And we tried on with two labels, ADO655 and 647N molecules, which are bright fluorescent molecules, and the thickness that we achieved is around 5 to 7 nanometers. We see a slight difference here in the thickness and the height of the bottom layer, and we attribute that to the properties of the molecule, or the fluorophore. ADO655 is a more hydrophilic dye and loves to be in the water around the bilayer, whereas ADO647N is a more hydrophobic dye and it sticks to the bilayers. So we have to still confirm this hypothesis with the additional measurements, but at least we believe that this is what we see. So far, I did not talk about the orientation of the single molecules. In principle, that plays also a very important role when you localize the molecules, even with a storm microscopy, or even here when you want to get the heights. So this is shown here. For example, if you have a dye of 1 nanosecond above your surface, it can be anywhere between these two limits in the height due to its different orientation. So it's important to know the orientation of your molecule if it is fixed, if the dipole is fixed in rotation. So we use defocused imaging. So this is a technique which has been established for a couple of years in our lab. What we do here is we shift the imaging plane of the camera and defocus it purposefully to blur the image. So it's contrast to what we actually do. We want to focus things, we defocus here. And as we know that the photons are emitted at different angles from the dipole, they hit now the plane of your camera at different positions, and you have a nice intensity pattern depending upon the orientation of your molecule, from which you can get the 3-dimensional orientation of your dipole. So we now combine this with a confocal microscope, and this is a slight modification that we do. We introduce a beam splitter which reflects 70% of the light on a camera, which can be defocused. And the rest of the part is focused on the lifetime detecting module. So the idea is that you scan, find the position of the molecules, and simultaneously record the lifetimes and the orientations to get both information to get the Z position. Just to give some numbers, we tried some test experiments. So here is a defocused image of a molecule and the lifetime curve. So if you bootstrap or you fit the molecule multiple times, 1,000 times, as in this case, you get the XY position, the distribution of the XY position of the molecule's position, and XY coordinates of the molecule's position, and also the error of estimating the theta, the out-of-plane angle. You can also do similar exercise on your lifetime curve, and you get an error for the lifetime estimation. Combined with these two, you will get the error of your axial resolution, and all the numbers are here almost a nanometer. So in principle, we have the potential to locate a molecule in 3 dimensions with nanometer accuracy with such simple experiments. The application idea would be in future the following. So let's say if you want to use measure a distance between 2 labeled molecules in your protein complex, the idea would be to put them on the surface, take a video of, so another condition is that the dyes should be blinking, so they are on and off, the cycles are in such a way that you can separate them. And because you also know the lifetime, because one of them is higher or one of them is lower in the z direction, you can sort out the frames with respect to with respect to which molecule the frame belongs to, and separately add them, as shown here. And you can then fit for the orientations of the 2 dye molecules, get the XY position, get the orientations, and then use the lifetime information to get the Z. In this way, you get the 3-dimensional positions of both the molecules, and you can estimate the distance. You will do that on multiple molecules, and get a distribution of the distance between the 2 sites of your interest. So we are now designing, with the help of our postdoc, Roman Sukarno, we are designing some probes, some test measurements. So this is a DNA paint MIET experiment where you have a DNA strand which is complementary to the docking stand here, which binds and unbinds, and we have 2 such strands laterally separated and vertically separated, and we want to test how far we can resolve the true distances with our experiments. So far I have been talking about static dye molecules, or dye molecules that are fixed in the sample. So the most useful, the use of a confocal microscope is also in studying conformational dynamics and dynamics. So for example, the conformational dynamics, for studying the conformational dynamics, the most used technique is FRET, Förster resonance energy transfer. We have 2 dye molecules, the acceptor and the donor. So the donor gives the energy to the acceptor, and the acceptor fluoresces. And usually this energy transfer is inverse powers R power 6, which means that you will measure only, this applies only for short distances, and you need 2 labels for measuring such conformational fluctuations in time. In contrast, we are introducing a new method based on a MIET, which is a dynaMIET, where you have now, let's say, 1 label on the end of the protein, and the other end of the protein is fixed or fitted, immobilized on the surface, and you have the conformational fluctuation in time. The relative intensity of your dye also varies proportional to the lifetime with the distance from the surface, so it increases. So as the fluctuation takes place, you have intensity fluctuations, which you can correlate to find the dynamics, the time of your the temporal dynamics of your protein or your polymer, for example. But instead of the FRET, the advantage here is that you need only 1 dye, because your dye acts as the acceptor of energy, and you can tune the distance up to 100 nanometers or even less with the help of ITO as your experimental design. So we are again going for some proof of principle experiments here. So this is a holiday junction of DNA, which has 2 conformation states. It is fitted to the spacer on top of the gold surface, and it has 2 conformation fluctuations, which depends upon the salt concentration. So these are some results from the FRET experiment in solution. We want to reproduce the same with the help of the dynaMIET experiments and see if we can reproduce this curve or not. Okay, so this also in principle works in the same way. So you go from point to point where the molecules are, and you collect the correlation till the molecule bleaches, and you go from point to point and average out for, let's say, 100 points. And this, for example, is the data from 1 successful point. And in principle, you average over all the points that you collect. So with that, I come to the last slide. We also are providing now tools for MIET imaging. The first here on the left, we see the MIET GUI, where you can feed in your lifetime data and feed in the parameters that you have, the thickness of your layers, the refractive indices, and the dye emission properties to calculate the lifetime calibration curve and to get from the lifetime the height distances and the height images, which I have shown in the presentation. Apart from that, an important recent development in the lab is the dead time correction. So when you do film measurements, the detectors and the electronics that count have dead time limitations, and which lead to a distortion when you measure at high count rates. So this figure, for example, was taken at a high laser excitation. This is the same cell on the left and the right. And if you see from the color difference, the lifetime shortens at this place because of the high intensity that we collect. We developed an algorithm to correct for such artifacts, and now we corrected this image, which is shown in here at the bottom, in principle, if you can see, the signal to noise here is much better because you collect more photons from the same positions. And this allows you to measure high count rate, fast film measurements, which is required for the MIET experiments or in general for any film community for film experiments. Okay, with this, I would like to come to the last and thank my group. Jörg, who is not here, but would have been happy to be here for all his crazy ideas and support. This is a MIET team. Alexei and Anna, who are standing right in the corner here, are responsible for the cell measurements that I've shown today. Daya is the main work behind the MIET GUI, the software. Roman is doing the DNA origami stuff that I presented, and the rest people are involved in the 3D MIET paint experiments and the dynaMIET experiments. Thank you for your attention. I would like to take the questions now.
Des questions? Questions? May I ask you one question? Could you come back to this setup for the 3D location of the molecules and explain it again for us? You know, not everybody is a specialist of this topic.
This
and
one or the previous one?
No, the one which gave this data, the setup itself.
Oh, the setup, okay.
Yes.
This one.
Yeah, this one.
So here I show, I'm sorry that I was really fast, but
yes,
here I show a confocal microscope. You have a linear polarized excitation. I mean, in principle, you don't care if it's linear polarized, which is reflected on the dichroic beam splitter and is focused on the sample. So it's a focus beam. And what we have is a sample scanning stage. So you scan the samples. And from the collection optics, you have the dichroic, which is passing the fluorescence light. You have a beam splitter, a non-polarizing beam splitter, which gets 70% of the light on the camera, which is the intensity camera here. And you can defocus this camera on a rail, so that you have this defocusing effect. And the rest 30% is focused through the pinhole, so the confocal part, onto the APDs. So this part, again, that gets the lifetime information, and this part gives you the 2D defocus image. You scan, you find the location of the molecules, you go from position to position on the molecules, and you record simultaneously the defocus image and the lifetime for the same molecule. In that way, you have the lifetime and the 3D orientation information.
Maxime.
Yes, well, congratulations for this impressive result. I wanted to ask you two things. So first, you have to scan, if I understand. So there's some sort of temporal dynamics is, if you want to look, for example, at the cells that are moving, is necessarily a bit limited or by the sequential acquisition that you have to do. So I don't know if there is any kind of solution for that.
For the cell measurements.
For example.
Okay.
And associated to that is that the fact that you always presented your biological structure, either, for example, the measurement on the membrane or maybe this DNA molecule that you attach to the surface, as almost immobile. So I would expect that, you know, the membrane is not immobile, it fluctuates, but
sure.
Since the amplitude of the distance that you can resolve are, you know, tiny, like it's beautiful, this 4 nanometer or 5 nanometer. I mean, I don't know how much would be the amplitude of this fluctuation, but I'd be surprised if they're much smaller than this. So how do you take that into account? So
first, for scanning, I mean, you now have rapid scanners, which have a dwell time of 2 microseconds per pixel. You're right. I mean,
you need enough photons.
Sure. So you're right. I mean, so there are also lifetime cameras now on the market, which can image life. So what you're measuring is the average height of your molecule during the scan. Of course, when you have a point scanning system, I mean, you lose. You have a different state when you have reached the end of the cell. But these lifetime cameras in the future, for example, will give you a much better or temporal average over the whole cell for the whole frame, the time duration that you expose to get the average height during this process. I mean, if you label enough, you can go down to 1 second or 5 second resolution. And during that time, I mean, relatively, for cells at least, you don't have so much of fluctuation in the cell. But if you want to record the temporal fluctuations at specific positions, then you can stay on this point, and then you can correlate because the intensity varies in time. So when you know where exactly you want to look at, for example, you have some complex which shows some dynamics in the microsecond, millisecond time range, then you can sit on a point and you can take a correlation in time. Then you will get from some microsecond to millisecond time scale all the information that you want.
Margaret Buckingham, just there.
I have a naive question from a developmental biologist. The beautiful pictures that you showed of the 3D phenomenon of EMT, for example, where you see the epithelial mesenchymal transition, this, I suppose, is done on single cells on a dish. Is that right?
So we did.
My question really is, what is the potential of using such techniques to look in tissues? That's to say, to hone down on the cells which are undergoing the phenomenon within a tissue explant or even within an embryo.
So we right now are measuring on the surface because our phenomena is surface limited. So the measurements we did for EMT are both on individual cell level and two, confluent layer, a monolayer of cells. So in both cases, we see the same behavior, temporal behavior of your fluctuations in the cell membrane. And the expansion, the expansion is, the change of the morphology is not so pronounced when you go to the monolayer level because they are squeezed. But however, the temporal fluctuations and the average height that they express on the surface is more or less the same. But if you want to go to deep tissue, I mean, we don't have so good resolving techniques in deep tissue, for example. That is a limitation. I mean, this is a surface-based effect, the metal-induced energy transfer. And that's the reason why we are at the moment restricted to the surface level. I mean, there are three-dimensional super resolving techniques, for example, multiplane imaging that we are also developing, but the resolution in that case cannot be higher, or I mean, it's not higher than 50 or 100 nanometers currently.
Maybe a last one. How does the fluctuation amplitude compare to the average distance in this experiment with cells?
I think I have a backup slide for that. Okay, maybe not. So the intensity, I mean, so if you see the lifetime modulation curve over the metal surface, it is pretty steep in some areas. So for gold, for example, between 40 and 50 nanometers, you can, that is the steepest here. Whereas it smoothens out. Yeah, whereas it smoothens out as you go higher. So this is the best range, and here you can resolve, at the moment we are trying to resolve 10 nanometers or 5 nanometer fluctuations in height in this realm. You can tune this, you can select a different metal layer, for example. You could go to if you could go to blue wavelengths, where the curve is much steeper, and then you can resolve for different distances from the surface at different level of distances of fluctuations.
No, that I understand, but my question was
the intensity.
Your experimental result.
The amplitude, yeah.
You know, what you measure, I mean, when it's in epithelial phase or when it's in mesenchymal phase, how do they compare, and how is the amplitude of fluctuation compared to the average?
I do not have data for that, but yeah. So the fluctuations here are certainly, so during the first stage, the fluctuations are higher, and during the transition state 1. After this, you have the dominance of the adhesion site, and the fluctuations go down. You mean the height?
It's small compared to the average.
Sure. So this is the error bars. Yeah, sorry. This is the error scale bars, and we measured up to 30 to 40 cells for each time point. And the fluctuations in the error bars, so they are less as compared to the actual height. Yeah, sorry. Yeah.
Thank you.